This video covers the Ctenophora or comb jellies. So we've covered these organisms, and the last group we covered were the cnidarians. And the ctenophores are sister group to the cnidarians, and a superficial look may make you think that they are very similar to a medusa form of a cnidarian, but they're actually quite a bit different when you look at them up close. So the comb jellies are represented by only about 200 species. They have a similar tissue level complexity to the cnidarians. They're diploblastic with an epidermis made up of ectoderm and a gastrodermis made up of endoderm. They also have a gelatinous material in between these. It's called collenchyma. It's basically just another form of mesenchyme. And they also lack a coelom. They're basically radially symmetrical, but in truth, because they tend to have a pair of tentacles, that makes really only one way that you can split them bilaterally symmetrical. But because they have an overall radial symmetry, we call that biradial symmetry. The only support they have is hydrostatic pressure and some of the support fibers that are actually in the collenchyma. They do have actually a fairly well-developed muscle tissue structure, more defined than what we saw even in the cnidarians. The muscle tissue is actually derived from the collenchyma itself, which differs from where it was derived, the two different locations, the epidermis and the gastrodermis in the cnidarians. And it consists of both very well-articulated longitudinal and circular fibers found in these sheets. Some of these muscle fibers actually have characteristics of smooth muscles and some striated muscles, so there's quite a bit of complexity here. Their nervous system really is built around a sensory structure called the apical sense organ. It's on the aboral end of the body, and it contains a central statolith. So if we're looking down on top of it here, we have this depression in the organism, and here we have this round statolith that's surrounded by these four long strands of sensory cilia. Each of these strands then leads to and branches to join these 8 ciliated comb plates, which are called teni. Now, the function of the apical sense organ is really twofold. One, the statolith helps to figure out the direction of gravity and movement, so it helps to maintain balance and orientation. But the strands that go to the tenae coordinate the cilial contractions in these comb rows, and so it actually allows for some coordinated directional movement. Both the apical sense organs and the tenae are synapomorphies for this group, so they identify an organism as a comb jelly if it has these two structures. They're generally pelagic, but again, they do have some limited directional mobility via their coordinated beating of the 8 teni, or the comb rows, hence they get the name the comb jellies. Most comb jellies are filter feeders. They capture zooplankton with their tentacles, and they have structures to help them capture this prey called colloblasts. These colloblasts are basically glue cells that when they rupture in contact with zooplankton, they create these sticky filaments to help capture that food, and then these tentacles can be drawn over the mouth to actually eat these captured food particles. Now, some internally in the gastrovascular cavity have a red pigment that covers this gastrovascular cavity, and it's been suggested that this is a way to hide the bioluminescent prey that they may have already captured. I don't know really what that's based on because, as we'll see in a little bit, the ctenophores themselves are bioluminescent. And one of the functions of that may be actually to attract prey. Once food is captured, as I mentioned, the tentacles are taken to the mouth where the food is deposited into the gastrovascular cavity. And the gastrovascular cavity is now a complete digestive tract. So here we have the mouth, and then we have the aboral end, where it has the 2 small anal pores. We see that there is quite a bit of branching of the gastrovascular cavity, and so most of the digestion occurs here extracellularly. And then because this is so highly branched throughout the body, there really isn't much need for a circulatory system to transport this material. The closest thing to a circulatory system is a series of cells called rosette cells, and they do help to move the digested material from pores in these gastrocavity canals to other parts of the body. But other than that, there is no circulatory system. And in similar vein, there's no respiratory, excretory, or temperature regulatory system. Gases are simply diffused across the epidermis and gastrodermis, and nitrogenous waste is the same. And again, these animals are ectothermic. It does appear that these animals are still capable of asexual reproduction through fission, and sexually reproducing. Most individuals are monoecious, so both male and female reproductive structures. And they can reproduce externally with fertilization. So they release their gametes into the environment, and then those fertilized zygotes have to take care of themselves. So this is a form of oviparity. As I mentioned, most of them are monoecious, but it turns out that most species have the capability of self-fertilization and cross-fertilization as well. Self-fertilization is relatively rare in animals because it has some genetic issues. Inbreeding has some negative genetic consequences in many cases. So again, most species are oviparous, but others do show ovoviviparity. And the gametes, when they are released into the environment, it's through the mouth. The eggs are centrolecithal, so this is something new. We haven't run across this yet. There is a concentration of yolk in the center part of the egg that then still undergoes holoblastic cleavage. But the cleavage is a little different. It shows some aspects of radial and some aspects of spiral patterns. So it's not clearly radial as we saw in the other groups. Gastrulation does result in the blastopore forming the mouth, however. The developing embryo turns into a sediped larval form that is used for dispersal. As far as how long these live, they do still have quite a bit of abilities to regenerate because of the retention of stem cells, but lifespans really aren't known. Sociality, there is none. They appear all to be solitary. Most ctenophores or comb jellies are bioluminescent. And the functions of this are not always clear, but two suggestions are that it may actually help them to escape predators if they're attacked, so they can flash that color to try to startle a predator while they escape, perhaps. But also it may help to actually attract and capture some of the zooplankton prey that they have if they are positive phototrophic. Comb jellies are key predators of zooplankton communities, and they do actually help to regulate the numbers of individuals in the zooplankton food webs. So much so that invasive species have had some pretty negative impacts on zooplankton communities in the southern parts of the Caspian Sea. And this has led to real problems with local fishing industries, and the reduction of the zooplankton has had a cascading effect, so that the zooplankton would have kind of kept some of the cyanobacteria populations in check, and it's released some toxic blooms associated with these cyanobacteria. So in review, the ctenophores or comb jellies are diploblastic. They don't have a body cavity. Pretty radial, but technically most of them are biradial in symmetry. They don't have a very firm skeletal system. It's just hydrostatic and fiber support in the parenchyma. They do have complex muscle tissues, however, with both smooth and striated muscles and longitudinal and circular muscles. The nervous system is centered around a coordinating apical sense organ that has statocysts that send out signals to the antennae for orientation and swimming movement coordination. But they don't have any osmoregulatory, excretory, circulatory, or respiratory systems. They're filter feeders, and they capture their food in tentacles that are lined by the sticky cell-producing colloblasts. They have a complete digestive tract with mouth and 2 anal pores. And then the eggs are centrolecithal with holoblastic cleavage, but variable cleavage patterns as far as radial and spiral. But the blastopore becomes the mouth. The larval form is called a sediped larval form. We talked about the potential ways that they use bioluminescence and how bioluminescence might be adaptive in different situations. And we talked about their role in eating zooplankton and how this can be problems when invasive species of ctenophore are introduced into areas where they're not native.